All right, so that was a good intro into rare disease. So understanding that anything I talk about today is I'm trying to get some very hard statistical problems, and explaining those in 30 minutes is very challenging, but I'll do my best. So I decided the best way to tackle lessons learned in designing clinical trials in rare disease is to go through several case studies and kind of highlight common themes that I found of things that we've done in designing trials in this space, and things that I believe allow us to be a little bit more successful than others in this area. So the first case study is in a disease called dominantly inherited Alzheimer's. This is a very rare version of Alzheimer's. It affects less than 1% of the population. In an individual who gets the genetic mutation, they will absolutely develop Alzheimer's, and they will likely develop it at an age of onset between 30 or 50, sort of around when their parent, who also had the genetic mutation, developed Alzheimer's. Within dominantly inherited Alzheimer's, we're involved in the dominantly inherited Alzheimer's network, which has a very large observational study following these individuals, as well as running a phase 2-3 platform trial in this area, meaning multiple therapies are coming into the trial and being tested to see if they can slow disease progression. So do they affect cognitive disease progression? Another example of designing clinical trial in rare disease is a disease called DIAN myopathy. This is also a very rare genetic muscle disease. It's categorized by slowly progressive muscle weakness, so it sort of starts in the ankles and moves its way up and into the body. You can see sort of MRI scans here of people at different stages of the disease. The muscle, it's believed that it occurs for a lack of production in sialic acid, and it's often caught when the subject or individual begins to trip, because their dorsiflex muscle will start to turn into fat and decline, and they can no longer lift their foot up enough to walk appropriately. And it's very slowly progressive, so this picture here to all the way this picture could be somewhere like 40 years, but it does end up, almost all of the muscles will completely decline and turn into fat. And so we're involved in developing a clinical trial in DIAN myopathy. It's a phase 2-3 trial. It's with individuals at the NIH. They also have a very nice natural history study in DIAN myopathy, and it's also with NeuroNext as well. And the goal there is to understand, does a treatment slow this sort of decline in muscle strength? And the last rare disease I'll talk about that we're interested in is FOP, fibrodysplasia ossificans progressiva. This is, again, a very rare genetic disorder, a connective tissue disease, and it causes the fibrous tissue to turn into bone. So this is a skeleton of an individual with this disease. And the median age of diagnosis is 5, so this occurs in very young individuals. The bone events, the FOP events, bone growth events, are brought on very rarely and sort of randomly. So when a person has an event is not quite easily predictable, and how much bone they'll grow is not quite predictable as well. If you can imagine a subject or an individual might go years without having an event, or may have many events in different areas of the body within the same year. Also, the amount of bone growth depends on where the event happens. If it happens in the hip, there might be a lot of bone growth, and if it happens, say, in the wrist, maybe less so. Interestingly, when we were originally trying to treat this disease, we were doing surgery to sort of remove the bone, and then realized that that was actually causing more damage, because for some reason when the tissue is stressed or damaged, it turns into bone. So it's actually causing more harm than good. So the goal here, again, is we're designing a phase 3 trial with a drug company to understand if there's a treatment, if their treatment is able to reduce the amount of bone growth in these individuals. So the previous talk was actually a good lead-in to this. I think really truly thinking about what's the best clinical endpoint or best primary endpoint to study is something that problem is magnified in rare disease when you have very few patients. So in all three of these, the issue truly is the heterogeneity in progression. People progress at very different times, at different rates in these diseases. In GNE myopathy, you might be actively progressing on the dorsiflex. One individual, another individual may be wheelchair bound and sort of actively progressing more on the upper extremities. So there's a large variability in key clinical endpoints within these populations, and different endpoints are affected at different stages of the disease. So it's not really clear what's the best primary endpoint to use. Common solutions to this, and what was alluded to earlier, is, let's just enroll a more homogeneous subset. Maybe in GNE myopathy, let's only enroll people who are actively declining sort of on the lower extremities, who can still walk a certain distance. Or enroll a large enough sample size to overcome this heterogeneity. Obviously, both of these are not feasible solutions in the rare disease setting. Obviously, if you try to enroll a more homogeneous subset, you're making a rare disease even more rare. And oftentimes we don't have large enough sample sizes to overcome the variability in some of these endpoints. So what I thought I would do then is talk about just briefly some overall themes that I've seen with these three designs and what can be helpful in common solution or some good solutions for rare disease. Across the board, in all three of these designs, we are blessed with very rich natural history studies that have been done before we got involved in the projects. And that allows us to, and I'll dive into this in a little bit, but that allows us to really understand the possible endpoints, to know what we're working with here. And so in each of these, we're able to sort of model disease progression, understand disease progression based on key clinical endpoints, and use that to appropriately design sort of evidence-based clinical trials. And then the second area I would say is more innovative design and analyses. So really in rare disease, you need to have designs and analysis methods that make the best use of all available data that you have. And I'll go into that a little bit more in detail as well. So the first point is natural history studies and why I truly think they're invaluable in rare disease for understanding several things. The first is, as I alluded to, that you can better understand the behavior of candidate primary endpoints. So a great example of this is in GNE myopathy. So as I said, when this slide was made, and I think now we're up to about 50 patients, but we have a really nice natural history study in GNE, about 38 patients at this time, and the follow-up ranges from about 0 to 32 months. And we have lots of measures being taken on these individuals over time. So one of them is 6-minute walk, which is a lot of the endpoint of choice for a lot of these types of studies. And another important one is a quantitative muscle assessment. So this actually is measuring the force you exude on each muscle in kilograms for every primary muscle group in the body. So we're able to look at the natural history data, and this is plotting individual trajectories of the natural history data over late the follow-up. This is for 6-minute walk. So you can see large variability in the baseline lengths that people can walk. And then over here I have plotted the change from baseline for these individuals on 6-minute walk. So we're trying to get an understanding, as was alluded to in the previous talk, of how sensitive this measure is to measure progression in a control, what will be a control patient. So with this natural history study, we are able to estimate sort of the sample size needed if you have a 1 or 2 year trial, and we calculated that you would need about 300 patients with a 2-year trial, which is just not feasible in this population. Also, if, and you'll see why I'm talking about this number later, but if you proposed 80% trial, 1-to-1 randomization, you would have about 30% power using this endpoint. This is another possible endpoint, and again, the next slide will make sense when I'm showing this specific one, but this is the upper extremity composite score. This is a sort of average of the muscle strengths of the upper extremities. It's within a subset, so it's within a subset of individuals who could walk a certain distance on the 6-meter walk. In this one as well, we saw that you would need, for a 2-year study, about 120 individuals, which again is not feasible in this rare disease. And if you did sort of a 1-year, 80-person trial, your power would be about 21%. So why do I keep focusing on this 1-to-1 80-person trial? In August of 2017, Ultragenyx announced the results from their phase 3 study. Their phase 3 study was an 80-person 1-to-1 trial for less than a year. And I would call this, based on Rob's talk earlier, a failed trial. So their trial did not demonstrate statistical significance. As you can see from this previous slide, we're actually predicting this before these results came out, that this trial was severely underpowered. And truly, this is very unfortunate in this area where no treatment exists for these individuals, and this trial is not answering the question of whether this treatment actually helps these individuals or not. So another way that we use natural history studies, other than just sort of looking at the different possible endpoints, is many of the trials that we design, almost I would say all of the trials that we design, we do clinical trial simulation. So we simulate the trials, we simulate subject level data based on likely assumptions of what that data would look like, and we look at very different trial designs, so boxes here. And we do this thousands of times to sort of understand what's the operating characteristics of our trial. So this is how we understand in more flexible, adaptive, innovative trials, what's the power of that trial and what are the operating characteristics of that trial. So natural history studies are truly an invaluable tool when it comes to clinical trial simulation. They allow us to come up with evidence-based and realistic virtual patient simulators, so how we simulate those patients and what they'll look like being enrolled in the trial. And this is really something that allows us to make sort of evidence-based decisions on key clinical trial questions that we might have, such as how many patients should I enroll, what's the length of follow-up, what analysis method should I use, and so on and so forth. So to give you just a quick, brief look into what those clinical trial simulations look like, or virtual patient simulators, I'll show an example with the DIAN trial. This is the dominantly inherited Alzheimer's. This is data from their observational study. And this was published, and in the papers that I put in their meetings for this presentation. The red dots here are people with the mutation. So these are people who will develop Alzheimer's. The black dots are non-carriers, so people who will not, or may develop sporadic Alzheimer's, but not dominantly inherited Alzheimer's. So you can sort of see on the x-axis, this is expected years to onset, and this is based on when the parent of this individual had onset, and also based on their specific mutation. And on the y-axis, this is cognitive decline, based on a cognitive composite score. And so you see this sort of waterfall effect of pretty rapid decline around an expected year of onset of 0 for the mutation carriers. So the question is, how quick is that decline? And we would really like to have some sort of disease progression model to understand that average decline over the course of the disease. So we built that model, and again, more details are in those papers I circulated, but sort of try to understand this average rate of decline as a function of estimated years of onset. Once we better understood that, we can simulate data from this model. And that's the data, the patient level data that will feed our clinical trial simulations. So trying to demonstrate this, for instance, this black curve is what came out of our disease progression model. This is the average across all the people as a function of expected years of onset of cognitive decline. Let's say we simulate one subject for a clinical trial. So this subject comes in, they were enrolled, they have an expected year of onset of 5. So they were actually 5 years post-onset, we believe. We also know there's variability in just sort of your inherent cognitive ability at baseline, so that's kind of captured by what we call a subject level random effect, and this subject has a negative effect, so they're slightly below average in terms of their cognitive ability when at a normal state. So we believe that this subject would follow, if they were a control subject, would follow this sort of black dotted line on average. However, they're actually a treated subject, so we think that there'll be some, under this assumption for the treatment, some slowing in the progression, and they'll follow this blue line. So we simulate with noise around this blue line. And so this is one patient who came into the trial, we followed them for 4 years, and this is what their data looked like. Again, another subject comes in. This one was 4 years post-onset. Also had sort of a negative random effect. This is a control subject, so we simulate them from this dotted line, and that's what their data looks like. So on and so forth. This is 10 subjects. This is their sort of values as a function of years since baseline. And here I've plotted the mean sort of values for the treatment and control. So we do start to see, even in this simulation, a difference between the treatment and control group at 4 years. This is a very quick walkthrough of a demonstration of how we actually simulate patients. So then this patient level data would go into our sort of design box and different boxes to answer different questions, and out would come an answer of, was this trial successful or not? And we do that thousands of times and report like how many times were the trials successful, what was the average sample size of the study, if it's an adaptive design, and different sort of operating characteristics of the design. So just to demonstrate that whole process and the overall operating characteristics, I'm showing an initial proposed design for the DIAN trial. This was proposed initially to be an 80-subject trial. As I mentioned, it's a platform trial, but I'll get to that a little bit later. But just imagine a single arm in the trial, single treatment, 80 subjects, randomized 3-to-1 treatment to control. We follow everyone for a max of 4 years, and the initial analysis method was a mixed model repeated measures. This is a common analysis to do when you have this sort of repeated measures, longitudinal data. This black curve is the power of our proposed trial as a function of true percent reduction in decline. So if you completely stop the disease, you have a very high power, almost 1. And if the drug is not effective, a low type 1 error, less than 2.5%. And this is really our targeted area. So this 3% was where we believe drugs will be, clinically significant effect, essentially. And what we saw with all this natural history data, all these fancy clinical trial simulations, is that proposed study was majorly underpowered, 20% power. So that leads us to design innovation. So what can we do now that we realize that our study is underpowered? So the first design innovation, and one that's common to all of these, is we do more innovative analysis methods. So all 3 studies have a Bayesian primary analysis. The DIAN network and DIAN myopathy have an analysis method that sort of takes into consideration and adjusts for disease severity. And you'll see that a common issue with the traditional analysis that's done in this area is that you get sort of a dilution of an effect due to subjects that are not expected to progress. So as I mentioned, you can't really just only enroll a homogeneous population that you expect to decline. You can't limit your sample size in a rare disease that way. So you'll be enrolling the whole population, and many of these, based on just the timing and their disease stage, they're not expected to progress over the course of the disease. So there's no hope that you'll see a change from baseline between treatment and control. So you see this sort of dilution of effect due to the people who aren't expected to progress. And also the mixed model repeated measures tests the effect at a single time point. So it tests it at 4 years. What's the difference between treatment and control at 4 years? We in DIAN and similarly in DIAN myopathy is developed a model that adjusts for EYO in the case of DIAN. In the case of GNE myopathy, it was this sort of latent disease age parameter. And it assumes sort of a proportional treatment effect at each EYO. So this is not a constant treatment effect. We don't think that the magnitude of the difference between treatment and control will be the same at each EYO and each time point, but proportional. So here, sort of if you enrolled someone at -5, if they're a control patient, they might progress and the difference between a control and a treatment patient would be about 0.125. You can see between the green and the black curve, you can slow the progression by 50%. Whereas someone enrolled later on, that absolute difference would be quite different. So not to go into too many details, again, this is in the papers that I've added for this talk, but this disease progression modification analysis, it uses all time points. So it's informed by the difference seen at 1 year, 2 years, 3 years, 4 years. And this is especially important, and where we gain a lot of power is that when we're enrolling in these slow rare diseases, enrollment can take quite a while, and we can have pretty long extended follow-up on individuals. So those individuals who enrolled at the beginning, they could actually be followed for 5, 6, 7 years at the end of the trial. And if we're not using an analysis method that incorporates that sort of extended follow-up, then really we're not using the data as best as we can. So this method allows you to estimate that treatment effect from extended follow-up. It allows you to better handle missing data and also like differential lengths of follow-up due to early interim analyses. So here you can see the same black curve as before, less than 20% power. Now just with this different analysis method, we came up with over 60% power for the same trial. So substantial increase in power by just using the data a bit more effectively and adjusting for disease severity in the analysis method. So another innovative aspect of the DIAN trial is it's a platform trial, as I mentioned. And so what's a platform trial, if you're not familiar? It's a trial, it also is sort of called a master protocol. So it has one protocol and enrolls multiple drugs. In this case, actually from very different drug companies. So you can imagine we have 2 drugs being enrolled into this trial, drug A and drug B. Both are enrolling 80 subjects, 3 to 1. So we have 60 treatment and 20 placebos for each drug. And this is also an adaptive design. So we actually have several interim analyses where either drug could stop for success or futility if it's not looking good. The way we're most effectively using the data in this situation is we're actually sharing the placebo data between the different drugs. So when we do an analysis on drug A, we're using the 60 treated subjects as well as the placebos from drug A and the placebos for drug B. So we're sharing this control information between the 2 drugs when doing the analyses. And this sharing in controls, or borrowed controls, takes us from a little bit above 60% power to nearly 90% power in our study. So you can see just with a better analysis method that adequately uses all of the data and sharing placebo data between 2 treatments, we're able to take the power of our study from less than 20% to above 90%. And because this is an adaptive design, we're actually able to have a chance to stop earlier than 4 years. So there's about a little less than 40% chance in this trial that we would stop at 2 years, stop a drug at 2 years early, a little bit above, like about 65% we would stop at 3 years, and then that 90% power to be successful at the end of 4 years. So in summary, really what I wanted to get across today is that natural history studies and clinical trial simulation, especially in rare disease, is truly invaluable for understanding what's the best endpoint to use, what's the best analysis method, and understanding key design questions you might have for your clinical trial. We were able to find out that the original DIAN power was less than 20%. And then also as well that in rare disease especially, we need better analysis methods that make use of the data, all available data, and adjust for sort of expected progression. And this allowed us in the DIAN case to go from a power less than 20% to above 80%. And this was similar in the GMD myopathy case. So in that case, we actually don't, that's a randomized controlled trial. There's no borrowed placebos or sharing of natural history data, but we were able to take that like about 21% power you were seeing on those previous slides and propose a 50-person trial, randomized 2-to-1, using sort of this disease modification analysis with multiple muscle measures that has a power over 80% as well. All of this comes with a word of caution for statisticians, in that we can make very complex analysis methods and models. We have no problem doing that, but to work closely with your clinicians to make sure that the models actually make sense. So all of these disease progression modification analyses wouldn't make sense if you thought you had a treatment that just gave sort of an initial symptomatic benefit and not sort of a disease modification, consistent benefit over the course of the disease. So just work closely with your investigator, make sure you're very clear about the assumptions if you're doing more of an innovative analysis, and make sure that those assumptions are plausible and believed. And because we're supposed to show dog pictures, and I'm an overachiever, I show my 2 dogs and my dog, and their handler here, my daughter. All right.
Thank you for a wonderful session.
Let me hand over to Dr. Wu.
I had 2 questions. The first one is, have you done the simulated patient model and active patient data from, say, a larger study? And the second one was the disease progression modification analysis. That was a really fascinating technique. I've always thought that we could better utilize all the data that we have. Why are we doing this in, non-rare diseases analysis?
Yeah, so we actually are doing it in non-rare disease as well. So we have another Alzheimer's trial through the EPAD consortium, so that's a European Prevention of Alzheimer's Dementia, and we're using a similar sort of slowing in the rate of decline proportional analysis as well. So we are starting to use it in others. I think regulatory is much more acceptable of innovative primary analysis methods in this rare situation where it's truly needed, and so that might be why we're not doing it in more cases in common disease. And what was your first question again?
Just about having done the simulated patient in live data, not from the rare disease, but from trying to simulate patient, taking the baseline data from, say, an Alzheimer's trial, not the rare one, but the regular Alzheimer's case, seeing if the simulations, then seeing what compared to the actual data result from the trial, see if it worked.
Oh, yes, we have done that as well. So a lot of times we'll look sort of at post-trial simulation. Is that kind of what you're getting at? Yeah, and see if, simulating under the actual data that came, if that sort of matched our simulation. That's the question.
Any other questions? Thank you.
I think Roger had one, but I'm a little afraid to tell you that Roger had a question.
Not a question, actually. Just when we're talking about simulation to test a trial, it's very tempting for clinicians to think you're trying to predict what the trial is going to do. And it's very useful to do the post-trial simulation for the actual data set, but it's really important for people to understand the goal of the trial is to have a trial that does something reasonable and gives you the right answer, regardless of what the truth turns out to be. So you want to simulate over a wide variety of things that might happen, you don't think will happen. If they happen, it would be horrible. If they happen, it would be great, to make sure no matter what the truth is, it performs well. Every time I get that question about, did you try this with real data? I worry that people are getting confused that we're trying to simulate what's going to happen in the future, and that's my hope.
I think Dr. Wu may be getting more at just sort of like that validation of the model to say, like, what's the residual? How well, if you looked at some prior data, were you able to predict, the progression in that? So